Hello everyone and welcome to the fourth lecture of EMSAT Achieve Biology series. Today we are gonna talk about the subsection 4 of section 1, which is identify and summarize the major cellular processes of breaking energy-rich molecules to obtain energy. So this is a unit which talks about biochemistry, and it is one of the most difficult section of the EMSAT biology. So I will be going a little slower on this and making multiple videos and break it into parts. Okay. The first thing under this that you have to understand, or know rather, is what is the difference between photosynthesis and cellular respiration, because this is something that we'll be discussing in the upcoming lessons. Photosynthesis and cellular respiration are exactly opposite processes. We all know what photosynthesis is, right? The plants, they take carbon dioxide and water. In the presence of sunlight, they make their own food, which is glucose, and they liberate oxygen. When you talk about cellular respiration is what happens in animals or in us. We take food or glucose, we inhale oxygen, and then we release carbon dioxide along with water. And cellular respiration gives us something very, very important, which is energy for our body. And this energy is in the form of ATP. Okay, so again, photosynthesis and cellular respiration are exactly opposite processes. Here, carbon dioxide and water reacts to make glucose and oxygen, and in cellular respiration, glucose and oxygen reacts to make carbon dioxide, water, and energy in the form of ATP. The next important concept is we are more discussing, this lecture will just be based on cellular respiration. Photosynthesis will be discussed in other lecture, not now, not in this lecture. Okay, so cellular respiration has two parts: aerobic respiration and anaerobic respiration. Aerobic is one which takes place in the presence of oxygen. Anaerobic is one which takes place in the absence of oxygen. So the reactants in this case are glucose and oxygen, but in case of anaerobic it's just glucose. We don't use oxygen in anaerobic processes, right? And what are the products? ATP, ATP, which is common to both. But in aerobic, we get water and carbon dioxide. But in anaerobic, we get fermented products. We will see lactic acid and ethanol, and along with carbon dioxide sometimes, which we see in yeast. What's the location? Aerobic respiration takes place in cytoplasm and mitochondria. The main site is mitochondria, because glycolysis is a common process for both aerobic and anaerobic respiration. We will see that. Glycolysis itself is an anaerobic process, but it's a common process for both aerobic and anaerobic, so we have put that in aerobic as well. Anaerobic totally takes place in the cytoplasm of a cell. Under aerobic respiration, as I said, for both anaerobic and aerobic, you have to know that glycolysis is common. But under aerobic, you have to learn about the Krebs cycle. Anaerobic, we talk about fermentation, lactic acid fermentation and ethanol fermentation or alcohol fermentation. Aerobic processes are actually very good. You know why? Because they liberate a large amount of energy. We get around 36 ATPs in aerobic respiration, but anaerobic respiration, you just get 2 ATPs out of it. Okay, so quickly going into the whole simple flow chart of what happens. So you have glucose. Now glucose is a 6-carbon molecule. It breaks into a 3-carbon molecule called pyruvate. This process is called glycolysis. Glycol means glucose, lysis means breaking down. So we break down glucose into pyruvate. This is a common process for both aerobic and anaerobic respiration, because if oxygen is available, pyruvate will make something out of Krebs cycle and electron transport chain. So this is part of your aerobic respiration. So when pyruvate is available, if oxygen is available, pyruvate will go to the mitochondria and start making stuff, which we will see what they make in Krebs cycle and an electron transport chain. But if oxygen is not available in the cell, then they will go for fermentation, which is lactic acid or ethyl alcohol. So let's start with glycolysis first. Please remember the glycolysis itself is a very big topic. We are not going to discuss the whole details of it. As I said, we will just be focusing on the key concepts which is important for your EMSAT exam. Okay, so glycolysis is an anaerobic process. That means it doesn't need oxygen. Okay, it takes place where in the cell? In the cytoplasm. What do we do in glycolysis? We break down glucose. Okay, glucose is a 6 carbon molecule. So ultimately what do we do? This is an intermediate product. With glycolysis is a long cycle. We are not going into the details of it. We are just interested in the reactant and the product here. So now when you break this glucose, you get 3C here, 3C's here. So you get 2 molecules of pyruvate, 1, 2, 3, 1, 2, 3, 6 carbon. Look, we break it here. So from 1 molecule of glucose, you get 2 molecules of pyruvate after the whole cycle of glycolysis. What is important is how many ATPs do you make in this cycle? We make 2 ATPs plus 2 ATPs, which is 4 ATP. But your net gain, the profit of ATP that is here, is only 2 ATP. Why? Because 1 plus 1, you are using 2 ATP to make 4 ATPs. Okay, so 4 minus 2, 2, 2 is your net gain. This is what is important for glycolysis. Sometimes they might ask you about the net gain of ATP. If they ask you about the net gain, it's 2. If they ask you about how many total ATPs you make in glycolysis, you will say it's 4. Okay. Now the next thing in this unit you have to know is about the Krebs cycle. The Krebs cycle is present in only eukaryotes. Who are eukaryotes? Those are cells which have a true nucleus. We are all eukaryotic cells, okay? We are eukaryotes. Who are prokaryotes? Bacteria. And we have a different unit which will discuss all that later on in some other lecture, not in this one. So just like glycolysis takes place in the cytoplasm, Krebs cycle takes place in the matrix of the mitochondria. Remember, mitochondria is a cell organelle. It is an organelle which is present in the cell, which makes energy for you. It's also called the powerhouse of the cell. Anyways, what do we do in the Krebs cycle? Remember, from glycolysis, what do we do in glycolysis? We break glucose into pyruvate, right? From 1 glucose, we get 2 molecules of pyruvate. Now, this pyruvate from glycolysis will go and make something called acetyl coenzyme. There's a long process involved here. We are not going into the details. Pyruvate is a 3 carbon molecule, remember? So out of this 3, 1 carbon goes out as carbon dioxide. The remaining 2 will make a compound called acetyl coenzyme A. Now this acetyl coenzyme A will start combining with a 4 carbon molecule, and there is a lot of chemical reactions happening here which we are not going to discuss in details. So acetyl coenzyme A combines with a 4 carbon compound to form something important which is called a citric acid. Citric acid is a 6 carbon molecule, and then there's a lot of things involved here, a lot of enzymes, a lot of products. So we'll just look for the basic thing. The 6 carbon citric acid molecule will make a 5 carbon molecule, and finally we go on producing a 4 carbon molecule which will again combine with this 2 carbon acetyl coenzyme A to make citric acid, and this cycle goes on and on and on. Okay, what is important is you should know what are you making out of it. So we are releasing 2 molecules of carbon dioxide, 1 and 2, and we are making 1 molecule of ATP, 3 molecules of NADH, and 1 molecule of FADH2. This is important. You should know the products that you're making. This is all about aerobic respiration. Remember, it also has electron transport chain, which will be discussed a little later. Again, from pyruvate you get your pyruvate from glycolysis. 1 carbon dioxide is released. From 3 to you make 2 acetyl coenzyme A, which combines with the 4 carbon molecule to make citric acid. Then they make a 5 carbon molecule. Lot of things happening. Finally, this cycle repeats itself. In the cycle, you produce some products What is that product? Remember, the glycolysis is just making two pyruvates for you, and you are using one pyruvate for each Krebs cycle, but you have two. So how many Krebs cycle will one glucose molecule will make? Two Krebs cycle. Okay. So if they talk about one Krebs cycle, you make two carbon dioxide molecule, one ATP, one FADH2, and three NADH. If they talk about two cycles, then you just double the amount. So now carbon dioxide is 4, ATP 2, FADH2 2, and NADH 6. Okay. Next thing you have to know is something called oxidative phosphorylation. Oxidative phosphorylation is a process which consists of two sub-processes. The first one is the electron transport chain, and the second one is chemiosmosis. These processes we will study again under photosynthesis in details, but right now I will just discuss the basic concepts and then we will stop. In the electron transport chain, what happens? Electron is being transported, is being transferred from one molecule to the other. And what does it do? When in this process, the energy keeps on releasing as one electron gets transferred to the other to form an electrochemical gradient. We will see all that under photosynthesis. And in chemiosmosis, we get energy stored in this gradient to make ATP. Okay. Coming quickly to the electron transport chain, the electron transport chain is the final stage of your aerobic respiration. And this one is not like Krebs cycle. Krebs cycle takes place in the matrix of mitochondria. Electron transport chain takes place in the inner mitochondrial membrane. Please make sure you know the structure of the mitochondria. Mitochondria is a double membrane structure. When we discuss cellular organelles, we will go in details about the structure. But for now, please remember glycolysis in the cytoplasm, Krebs cycle in the matrix of the mitochondria, and electron transport chain takes place in the inner membrane of the mitochondria. The inner membrane of the mitochondria is thrown in a lot of folds, and this fold is called a cristae. And what is the purpose of it? Is to increase the surface area for the electron transport chain. What happens in the electron transport chain is a long chain again, where you have a lot of electron acceptors. What is the basic thing it does? Oxygen is the final electron acceptor. So it's the last one in the series. It accepts your electrons and picks up the protons to form something which is called the water molecule. This will, as I said, we will discuss in details in the photosynthesis chapter. For this unit, I will stop here. In the next lecture, I will start with anaerobic respiration, which is fermentation. Thank you for listening. Please give your comment and feedback, which will help me to improve my lecture. And what do you want in the lecture? What extra things? Please let me know. Thank you so much.